Hello, everybody. I'm Gratiela Gradisteanu from University of Bucharest. I'm a biochemist by formation, but by heart, I'm a microbiologist. And today I'm going to talk to you about several aspects related to the gut microbiota. And we know we live in the microbiome era. We hear a lot of information about how important the microbiota is for health and on how to have a certain diet and what to do in order to have a good microbiota and a good status of health. And also, it's a very hot research subject, and it has really flourished in the last decade. And I'm lucky to say that I have been working in this field since 2012, and it was also part of my PhD. And I'm going to discuss about some of these results related to the microbiome from my research, and also about our current projects that we are doing in the University of Bucharest related to the microbiome. And it's really important because we are more microbe than human. Basically, all these microbes that live in and on us are more numerous than the cells of our body, and they accomplish multiple functions. They can help digestion, they synthesize vitamins, they are involved in metabolism, they guide immune system development, and they fight off pathogens. And who are they really? And I'm going to start about talking about the bacteria in a simplistic way. In a nutshell, we can say we have the good bacteria, the friendly bacteria, such as lactobacilli and bifidobacterium, and we hear about them all the time, even when we look at TV and we see some commercial about some yogurt containing these microorganisms. And we have, on the other hand, the unfriendly bacteria, the pathogens. Some examples are shown here, such as Helicobacter, Shigella, Salmonella, Campylobacter. And on top of that, we have the pathobionts, the bacteria which are somewhere in the middle, because normally they are good bacteria, beneficial bacteria, but in certain conditions, after a disease, after antibiotic treatment, they can become harmful for the host and they can promote inflammation of the gastrointestinal tract. And what else do we know about this microbiome? We know that we have 25 phyla with 2,000 genera and around 5,000 species. And all of these have more than 300 million genes. And moreover, we have bacteria being discovered on a regular basis. And there is really a very dynamic field of research. And on top of that, there are a lot of things we don't even know, we don't know. So it really is an amazing field of research. And these tiny beings have a big impact on us. So as I said before, they are involved in metabolism. They can synthesize various vitamins. They can synthesize short-chain fatty acids such as acetate, butyrate, and propionate. And they are really important for the development of a good intestinal tract. And they are a source of nutrients for the colonocytes. They have immunomodulatory activity. They can prevent inflammation. They are really important for immune system development and for the development of Th17 immune cells, for instance. And importantly, from my point of view as a microbiologist, they are involved in anti-infectious immunity. They can compete with pathogens, and they do this by competing for nutrients. They can produce different antimicrobials such as reactive oxygen species, lactic acids, or bacteriocins, and they can also block the adhesion sites for the pathogens. And this is a very, very dynamic system. Initially, it was thought that the placenta was sterile, but actually in the last years, it was shown that even in the placenta, we have some actinobacteria. Once the baby is born, it's really important the mode of delivery, because if the baby is naturally born, it will be colonized by lactobacilli from the vaginal microbiota of the mother. Whereas if the baby is born by C-section, then it will get colonized from all the microbes that exist in the hospital environment, mostly Staphylococci and Corynebacterium. And initially, we have a lot of Enterobacteria in the microbiota, then we get an increased diversity. Basically, in the first 2-3 years of life, that's when the microbiota gets settled. And really, it will resemble the microbiota of the adult. And whatever we do, it will influence the microbiota one way or another. And how we can influence it? Well, by taking antibiotics, by having a certain diet, and also by taking probiotics, prebiotics, and so on. And it's also very complicated because here in the gut, we do not have only bacteria, we also have fungi and we also have viruses. So we have also two additional fields that have emerged in the last years, such as the microbiome and the virome. And in the microbiome, we see here that we have species such as Aspergillus, Candida, Fusarium, Mucor, and Saccharomyces. And there are currently studies working to decipher the microbiome signatures in different diseases. And it's a little bit more difficult because the techniques are not advanced that much compared to the analysis of the bacteria. And also we have the virome, that's the total amount of the viruses that exist. And here we have microviridae and caudoviriales mostly as part of the virome. And of course, now since we have this pandemic, it's really important to study the virome as well, because who knows, maybe we have some members of the virome which can influence the impact of COVID-19. And how do we actually look at this microbiota? For many, many years, people were just cultivating this bacteria. But really, this is just looking at the tip of the iceberg, because most of these bacteria are not cultivatable. So we have a lot of techniques, molecular biology techniques, that we can use to analyze these microorganisms. So we can do a real-time PCR, and we can have a look at various bacterial populations by looking at their 16S ribosomal RNA. We can do some sequencing, or we can do metagenomics, looking at all the genes from these microorganisms, or metabolomics. And in this case, we look at the various metabolites that this bacteria produce and their impact on the host, and also at metatranscriptomics and metaproteomics. And in this case, we look at various transcripts or proteins. And how do we change this microbiota? Well, here we have a wide array of ways on how to modify the microbiota. In a simple way, we can change the diet, and that has been done in many, many ways. We know that the Mediterranean diet is really important because it's high in fiber, and it will promote development of a good microbiota. Probiotics have been on the stage for many years. We have the lactobacilli, the bifidobacteria, and also some Saccharomyces, which can be probiotics. And on top of that, we have the prebiotics, which are molecules which, given to the host, will promote development of the probiotics. And prebiotics are inulin or fructooligosaccharides and galactosaccharides. And when we use both of them, both the prebiotic and the probiotic, we have what is called a symbiotic. On top of that, we can use postbiotics. These are small molecules which can promote a good health for the host. For instance, in this case, we can use small peptides or teichoic acids or different polysaccharides, which again are good for the host microbiota. We can use antibiotics to change the microbiota, but not in a good way. And I'm going to show you some of my data regarding this. And also we can have bacteriophages, which are viruses of the bacteria, and they can also trigger changes in the microbiota. We have nanotechnological approaches, which can be used again to target the microbiota, or bacterial cocktails. As the name says, we have a multiple mixture of different microorganisms that can be administered in order to promote changes in the microbiota. Or we can use the fecal matter transplant, which is currently used in the clinic in patients with Clostridium difficile infection. That's a good way to actually cure this patient, because they are receiving the microbiota from a healthy individual, and that's how they get rid of the nasty infection with Clostridium difficile. And how it all started for me, as I said, I'm a product of University of Bucharest. I'm a biochemist, and I did my bachelor degree in the lab of Professor Carmen Chifiriuc, where I worked with Vibrio. This is a gut pathogen, and I was looking at the way how different strains of Vibrio, Vibrio fischeri and Vibrio parahemolyticus were responding to changes in the environment, such as pH and temperature and the heat shock proteins they produce under stressful conditions. And then I moved on to do an Erasmus stage in Greece at Democritus University of Thrace. And there I was working with enteropathogenic E. coli, and I set up some techniques on how to detect this nasty pathogen again from various sources, mostly from different types of food and environmental samples. And then I moved on to do my PhD at University College Dublin, and I started working with Campylobacter jejuni, again, another gut pathogen, and I was trying to set up an animal model, and it was really hard. That was my first year of the PhD, because it was not working, because normally mice are not human and they do not get colonized by this pathogen. So I had to switch my field of research, and that's how I reached the field of the microbiota, and I'm gonna talk about that right now. So I worked in the field in the lab of Ulla Knaus. She's a researcher now. She's in Ireland. She did a lot of research in San Diego, the Scripps Institute, and I joined her lab when she came to Ireland. And my co-supervisor was Billy Burke. He's a gastroenterologist, so he also guided some of my research because we were also very close to the clinical sites, and he works with inflammatory bowel disease. So he was also interested for some of the aspects that are related to our research. And our research was published in Cell Host and Microbes some years ago. And the main field of activity were these enzymes called NADPH oxidases. They produce reactive oxygen species. They're a family of enzymes. The most important one is NOx2, or the phagocyte oxidase, because it really is involved in producing reactive oxygen species when we have an infection. And in order for it to be functional, you have all these complex proteins, making this complex of activity, and that's how we produce reactive oxygen species. But we do not have oxidases only in the immune cells. We have it also in the gut, and we were also interested at this NADPH oxidase, NOx1. We also have NOx3. This is involved in multiple functions, including perception of gravity, and its activity is in the inner ear. We have NOx4. It's a little bit different in terms of structure, but it also produces reactive oxygen species and is found in the kidney and in the heart. We have NOx5. It's completely different because it requires calcium for activity and does not have all these regulatory subunits. And the duoxes, duox1 and duox2, they are found in mucosal tissues of the respiratory tract and of the gastrointestinal tract. What I did was to create new animal models to study these NADPH oxidases. So, as I said, most of these oxidases require a lot of proteins to make this complex in order to produce reactive oxygen species, and an important protein for most of them is the P22 protein. And it's important for the proper activity of NOx1, NOx2, NOx3, and NOx4. And what I did was to make a knockout mouse, which does not have this protein at all. So basically, the P22 knockout mouse will not have NOx2 functioning in the phagocytes and will not have NOx1 and NOx4 working in the epithelium. Mostly we were interested in the gastrointestinal tract epithelium. But of course, this is a knockout mouse. And when you do a constitutive full knockout mouse, you have a lot of off-target effects. And we wanted to do also a conditional knockout mouse. And in this case, we targeted the same protein, but in this case, we targeted only in the intestinal epithelial cell. That's why we have this mark here. It's a P22 delta IEC. So it's a conditional knockout mouse in the intestinal epithelium, which does not have these oxidases working, but the phagocytes are just fine. And of course, we needed the controls, which are the wild-type mice, which have all these oxidases nice and working and functional, and the NOx2 knockout mice, which does not, of course, NOx2, but will have the other oxidases working. And what we wanted to do in this case, well, we forgot about the Campylobacter because that was not working at all. And it's still not working for a lot of researchers around the world because of the microbiota of the mice. And we moved on to Citrobacter rodentium. And this is a natural pathogen of the mouse, and it mimics a lot what happens when we have an infection with enterohemorrhagic E. coli and enteropathogenic E. coli. And normally we would expect that this knockout mouse would get really sick because it does not have all these phagocytes, it does not produce reactive oxygen species, and we were even thinking, well, we give this pathogen and the mouse will die. But actually the opposite happened. And it was a little bit difficult because you're a PhD student and you say these things and people don't believe you and they look really weird at you. Well, how does this happen? So I had to do this experiment multiple times and bring a lot of proof to show that this is really the case, because the mice were not losing weight at all when they were getting the Citrobacter infection. They had low pathology scores, and we have those at 3 days and 6 days post-infection. They have low colonization, and look how different they are from the NOX2 knockout mice and the wild-type mouse. And even if we look here, the gastrointestinal tract, it looks very much similar to the one of a non-infected mouse, whereas in the wild-type mouse, we have inflammation of the cecum, and here in the NOX2 knockout mice, we have high inflammatory infiltrate in the cecum, as shown here. And we did some histology as well, and we see basically the same thing. These are not the non-infected mice. And when we get Citrobacter rodentium infection, we have a hyperplasia of the colon crypts. This is typical feature of this infection. And when we see that, we know that the pathogen has colonized and has triggered some changes in the intestinal epithelium of these mice. And we see that also in the NOX2 knockout mouse. But when we look at the P22 knockout mouse, we don't see anything at all. It really looks like an uninfected mouse. So, okay, it looks like this phenotype does not come from NOX2 because NOX2 really resembles the wild-type mouse. So we did another infection with the P22 conditional knockout mouse. So the mouse which has P22 inactivated in the colon. And we see again the same protection. The mice do not lose weight. They have very low colonization. And see here, each dot here represents a different mouse. And again, low pathology scores 3 days and 6 days post-infection. We look at the H&E staining of the colon sections. We have here the uninfected mouse. We have hyperplasia in the wild-type mouse, whereas in the delta EC mouse, we do not see that at all. And we see really it resembles what happens in the uninfected mouse. So we had this protection phenotype. Okay, so where does this protection come from? And in order to see where does it come from, we wanted to get rid of the protection. So we did this in using several methods. So first of all, we wanted to see, well, if we change the diet of the mouse, well, do they get protected again or they lose the protection? And for this, we customize the diet in the mouse, which mimics very much what happens when people eat a lot of McDonald's and junk food. And we have this Western diet in mice, which has a lot of fat, it has cholesterol, and it has a lot of sucrose. Actually, 34% of the diet is sucrose. We didn't change that much the amount of proteins. We look also how this diet affects the metabolism of the mice and how it will impact the infection susceptibility. And how we actually did this in the lab was to do a prenatal Western diet administration. We had the females that were pregnant, and 7 days after the conception, they were given this diet. And the mice that were born from these females all the time received the Western diet and also during infection. And if we look at them, we don't see that much difference. Well, they are a little bit heavier, and if you look at that, they are a little bit chubby like that. But actually, when you look inside of them, you see a lot of changes, and I'm going to show you that in the next slides. And we have some changes in the metabolome, which are induced by the Western diet, and we did this in a collaboration in UCD using NMR. And we have here the short chain fatty acids that I mentioned at the beginning of the presentations. They are really important for the host immunity, especially butyrate, because that's a source of nutrients for the colonocytes. And once we give the Western diet, both the wild type and the knockout mouse have decreased level of butyrate, decreased level of acetate, and also some decrease in the propionate levels. We have analyzed other metabolites as well. Lactate was severely affected by the Western diet, and I'm going to talk about this a little bit later and tell you why is this so important. And succinate was also decreased, and we also have high levels of taurine, especially in the knockout mouse, because this taurine depends a lot on whether the diet has high levels of fat, and we see that here clearly shown. And when we do the infection, well, of course, our hunch came true, because once we give a different diet and potentially we alter the microbiota of these mice, we lose the protection. And we have here some slides showing that we increase the intestinal permeability in these mice, and we also have changes in how the colon looks like. And we have here smaller colon crypts, and we have low mucus production. And also, as I mentioned, we do not see any difference at all now in the wild type and in the P22 conditional knockout mouse. See, same level of colonization, the mice start losing weight, and we have hyperplasia both in the wild type and in the knockout mouse. And well, this was not enough. So we did another experiment to change the protection phenotype. And in this case, we gave antibiotics, because antibiotics also trigger changes in the microbiome. And we did this by giving antibiotics both in the drinking water. We give ampicillin and ciprofloxacin, and by oral gavage. So we reach directly the stomach of these mice. And we gave amphotericin, metronidazole, vancomycin, and neomycin. We gave daily antibiotics to the mice for 2 weeks. Then we have a period of 1 week of recovery. And then the mice receive Citrobacter rodentium infection. And we know that the experiment worked, because we have this megacecum. This is indicative of a sterile gut. And look what happens when we abolish the microbiota using antibiotics. So we have the mice actually being a lot worse. These are the P22 knockout mice shown in red, which actually start dying from the infection. And they have dissemination of the pathogen to the spleen and to the cardiac blood in a similar way to the to the NOX2 knockout mouse, whereas the P22 conditional mouse resembles a lot the wild type mouse. And in terms of colonization, we did not see any difference, but we did have significantly higher levels of Citrobacter rodentium colonization when we abolished the microbiota with antibiotics. And we did another experiment. Well, what if we do fecal matter transplant to the wild type? Can we protect the wild type? So for this experiment, we used only wild type mice, which were depleted by their microbiota, again, using the same experiment as shown before, by gavage of antibiotics and antibiotics in the drinking water. Then we have the recovery period, and then we have the fecal transplant, in which a wild type mouse receives a wild type microbiota, a P22 knockout microbiota, and a P22 delta IEC microbiota. And what we see is, yes, when the wild type mouse receives the protective microbiota, it will be protected. See, whether it comes from a P22 conditional knockout mouse or a P22 knockout mouse. And we have it shown here, the colonization data and the histology data. So we have hyperplasia when the mouse receives another wild type microbiota. When it receives P22 knockout microbiota, we do not see changes in the histology of the colon. So what are these mechanisms of protection? By now, we had an idea that possibly it's the microbiota, but we also looked at the mucus, because the colonocytes are covered by this mucus layer, which has actually two parts. It has the inner mucus and the outer mucus, and in the outer mucus, that's where the microbiota resides. In the inner mucus, it should be sterile. And we did different stainings. We did here a staining for MUC2, that's the main mucin of the colon in mice, and we did not see changes between the wild type and the knockout mouse. And we did a pus Alcian blue staining, and you have here the inner mucus layer, which is similar between the mice. So again, this was another proof that this protection was coming from the microbiota. And then we had a collaboration with a lab with David Goodman in Canada, and he helped us sequence this microbiome. And for that, we chose the SECOM, because that's the site where most of our experiments were done, particularly the experiments regarding the colonization with Citrobacter. We did DNA isolation from the cecum of all these mice, and we sent off the DNA for sequencing to Canada, and we got the results back, and those made us really, really happy. We have here the main phyla of the microbiota, and these are the mice. Basically, each square is a different mouse. And we have here the phyla Actinobacteria, Bacteroidetes, Deferibacteres, Firmicutes, Proteobacteria, Tenelicutus, Verrucomicrobia, and the TM7. And basically, if you look at that, we don't see big differences, but the differences emerge once we look in the smaller bacterial groups. And we have here something amazing. At that time, it was really amazing for us because we see that these knockout mice, by default, they are born with a microbiota, which is enriched in probiotics, such as Bifidobacterium and Lactobacillus. And around this lactobacilli, the most important one was Lactobacillus reuteri. So clearly now this was the eureka moment that these mice have indeed a modified microbiota, and that's what protects them against infection. And we looked again at those mice that were receiving the Western diet, and we see that the Western diet triggers changes in the microbiota, causes changes in Ruminococcus, in Bacteroides, and the Sulfovibrio, Bilophila, and the Sulfovibria. All of these are datobionts, so those bacteria which can trigger changes and can cause inflammation in the gastrointestinal tract. And importantly, we looked at this little guy here, Lactobacillus reuteri, which was clearly affected by Western diet. So in normal diet condition, we have in the wild type mouse a lot of Lactobacillus reuteri. If the mice get this Western diet, then we do not see that change anymore. And we looked more at Lactobacillus reuteri because we see this change in the microbiota, and we wanted to have a mechanism, and I'm gonna present a little bit of this mechanism. Actually, the paper is more complex than that, but we're going to focus more on the Lactobacillus reuteri. And what we know about this lactobacillus is that it produces a lot of reactive oxygen species. So basically, we have a host which does not produce reactive oxygen species. And in this host, we have an enrichment of bacteria which can produce this reactive oxygen species for the host. And how does this actually work? We know that Citrobacter rodentium is very similar to enteropathogenic E. coli, and it needs this actin pedestal in order to infect its host. And formation of this actin pedestal from the cell host is regulated by a wide array of genes. I circled here the LAIR gene and the ESCN genes because we focused on these two, particularly the LAIR gene, which is the master regulator of virulence in Citrobacter. And what we see is that when we co-cultivate Citrobacter with this lactobacilli, we have changes in virulence, and we use the wild-type lactobacillus, which produces reactive oxygen species, and we have a mutant lactobacillus. We got this one from Nestle, which does not produce reactive oxygen species at all. And what we see is when we co-cultivate the Citrobacter with the lactobacillus, when the lactobacillus does not produce ROS, we have an increased virulence, we increased expression of the LAIR gene in Citrobacter. And we did that also in vivo. So we have the mice which were colonized with this lactobacilli. If the mice get colonized with the good lactobacillus, which produces reactive oxygen species, they have low levels of Citrobacter. And when they get the mutant, they have increased colonization with Citrobacter. And we look at the Citrobacter coming from those mice, and we have here decreased expression of the LAIR gene when we have a lactobacillus which produces ROS, and decreased expression of the ESCN, and the opposite in case of the mutant lactobacillus. And we also looked at the Citrobacter from two different sides. We looked at the Citrobacter that is in the lumen of the gastrointestinal tract and the one which is adherent to the tissue. And we see the same thing, that the lactobacillus, which we recover from the knockout mouse, will have decreased LAIR expression and decreased ESCN, as well as the one the adherent one, we have low expression of LAIR and the ESCN in the P22 knockout mouse, and the opposite for the Citrobacter recovered from wild-type hosts. And with this, we published our research in Cell Host and Microbe, and this was actually, they asked the graphical abstract for us, and we made the cover of Cell Host and Microbe, and this was actually drawn by my little brother, who is a graphic designer, and it actually shows here that the Lactobacillus knocks out the Citrobacter by using reactive oxygen species. And this was my work in University College Dublin, and then I came back to University of Bucharest as a research fellow. And here my attention was triggered by the human microbiota, because of course it's nice to work with mice, but our aim is to look at the human microbiota. And we know that the microbiota is connected to a lot of diseases, from diabetes, insulin resistance, obesity, allergies, liver disease, cardiovascular disease. We also have a gut-brain axis. So the metabolites produced by the bacteria can influence the behavior of the host, and they can be involved in depression, anxiety, and autism. And really, the research field is enormous. I mean, there are so many papers published every day about different microbiome signatures associated to various diseases. And this entire connection is influenced by the lifestyle of the host and the diet they have and their genetic susceptibility and their immune system. So it's a really complex equation that we have to solve. And when we have changes in the microbiota, we have what is called dysbiosis. And in case of a normal microbiota, we have a lot of anaerobic bacteria and some facultative anaerobes. Once the dysbiosis occurs in various diseases, we have a reduction of the normal anaerobes and we have an increase in facultative anaerobes, because once we have inflammation, we have increased oxygen at the level of the colon, for instance, because we have influx of immune cells. And that's when those pathobionts mentioned by me at the beginning of the presentation starts to flourish. So we have an increase of pathobionts, and we have also a fungal overgrowth, and we lose the beneficial bacteria, such as bifidobacteria and lactic acid bacteria. And we also published some papers regarding the microbiome in various diseases. I'm going to give you some example here. For instance, in colon cancer, we have the healthy microbiome, which is composed of a lot of Faecalibacterium and Ruminococcus, whereas in patients with different types of colorectal cancer, we have Fusobacterium, Enterococcus, a lot of Escherichia and Bacteroides, and we have increased gut permeability, and we have a lot of influx of immune cells. And similarly, in other diseases, we have a similar pattern of the microbiota, for instance, in inflammatory bowel disease. And my main area of focus is diabetes and the microbiome patterns in diabetes. We know that there's a link between diabetes and the microbiota. If we refer to type 2 diabetes, we know that if we have a diet which is high in sugar, high in fat, then we will end up having type 2 diabetes. And in case of type 1 diabetes, there are multiple factors that can trigger this disease. One of them, of course, is the genetic susceptibility. We also have what is called the hygiene hypothesis, where we have children from developed countries which live in very, very clean condition. They're not exposed to dirt, and they get autoimmune diseases, particularly allergies, asthma, and type 1 diabetes. But of course, there's still a lot of debate on this microbiome diabetes connection, because we don't know which one came first, actually. So there's still a lot of debate in the field at the moment regarding this. And I chose this topic of research because Romania is also on second place in Europe regarding the prevalence of type 2 diabetes. And it's a nasty disease because it's associated with a lot of complications, such as retinopathy, heart attack, cataracts, and diabetic foot nephropathy, and so on. And we know that in case of the healthy microbiome, we have out of Pircalabioru, we have Akkermansia, Lacnospiraceae, Veillonella, and Clostridiales. And in both type 1 and type 2 diabetes, we have increased gut permeability. So basically, we have these holes in the epithelium, which will trigger inflammation of the colon. And we have a lot of Enterobacteriaceae here. We have Streptococcus, we have a lot of Bacteroides. That's what most of the studies show, regardless of the type of diabetes, that we have Bacteroides in these individuals, and Intestinibacter in case of type 2 diabetes. So, of course, if you want to work with patients, you need samples. So what I did was to create my network of clinicians. So basically, I went to various hospitals. I told them who I am and what I know to do and what I want to do. And that's how I made this network of clinicians. Those are the ones I currently work on, and I received samples. There are actually more of them, but because we have the pandemic now, I don't have all the samples to actually do the experiments. So these are the ongoing projects at the moment. So we have Amalia from Elias Hospital, who works on new onset type 1 diabetes and childhood obesity. We have Dr. Anca Pontea-Stoian, who is the vice president of the Romanian Association of Diabetes, and she provides us samples from patients with type 2 diabetes and cardiovascular complications. We have Octavian, who is particularly interested in diabetic nephropathy, and we have at the moment, I'm working with one of his PhD students to analyze the microbiome in diabetic nephropathy, and Madalina Mușat, who works at College Institute of Endocrinology, and she provides us samples from patients with metabolic syndrome and type 2 diabetes. And of course, it was hard in the beginning because we did not have funding, so we had to do a pilot study. And we did a pilot study in using patients with type 2 diabetes and chronic kidney disease, and we used real-time PCR to analyze their microbiome. So we received the samples, we extracted the DNA, and we did real-time PCR, and we looked here at the total sequences of bacteria. We did not find significant differences, and we looked at the main phyla, Bacteroidetes and Firmicutes, and there was a slight increase in Firmicutes, but this was not statistically significant because we actually used only 14 patients here. It was a pilot study, as I mentioned before. And we have here other bacterial populations which we analyze. We have here Bacteroides porphyromonas prevotella. We have a small increase in the type 2 diabetes group, no changes in Clostridium coccoides. We have higher Clostridium leptum and no changes in lactobacilli. Strangely enough, we found Fecalibacterium prausnitzii higher in the type 2 diabetes group, and that was a little bit unexpected, but probably it's due to the fact that we had a small population under analysis. But what was interesting, and it was statistically significant, was the fact that we had an increase in pathobionts in case of the type 2 diabetes patients, where we had Turicibacter highly expressed in case of the diabetic patients and Enterobacteriaceae as well. And we, in parallel, did another microbiome pilot study with patients with metabolic syndrome, again with the qPCR. We do not see changes in EU bacteria, but when we look at various phyla, we see low actinobacteria in the metabolic syndrome group. We see more gamma proteobacteria, and these are the phyla that are composed of those Enterobacteriaceae, which are pathobionts. And we did not find changes in Firmicutes or Verrucomicrobia. And we look here, we see that we have higher levels of Enterobacteriaceae in the patients with metabolic syndrome. So again, we have higher levels of pathobionts and Turicibacter. So these are different patients, different hospitals, and we see the same pattern. So this probably is the real case of what's happening. And we have significantly higher Clostridium coccoides in the metabolic syndrome group, and we have levels of Clostridium leptum, again, higher in the metabolic syndrome group. We did not find changes in the Bacteroides though, and we have higher levels of Ruminococcus. And what was important was to also look at those bacteria which are producing butyrate, because I kept mentioning, well, these short chain fatty acids are really important, particularly butyrate. And they are important in patients with metabolic syndrome because they can be involved in decreasing blood pressure. And we see low levels of Fecalibacterium prausnitzii and Butyriciococcus, again, in the case of the metabolic syndrome patients. And what was the next logical step was to move on to sequencing this microbiota, because that will give us a lot more information. And we increased our patient numbers. So we got 40 metabolic syndrome patients, and among those 28 also had type 2 diabetes and 30 healthy controls. And for that, we extracted the DNA, and we established another collaboration with the London School of Medicine and of Tropical Medicine and Hygiene. And they did the Illumina sequencing for us. And we see here each bar that we have here, each multicolored bar represents a different patient. Here we have the metabolic syndrome patient and the control healthy individuals. What we see is that most of the individuals have Fecalibacterium as the main part of their microbiome. We have higher bacteroides in case of the metabolic syndrome patient. And we lose some species in case of the metabolic syndrome. So we have, for instance, Succini vibrio, which produces succinate, another metabolite important for the host, and we lose those completely in case of the patients with metabolic syndrome. We also did, with my collaborator, Ozan Kungdogdu from London, a lot of statistical analysis, and we are also working at the moment on this data to see how the different treatments these patients receive will affect their microbiota. And we also are doing some statistical and bioinformatical analysis to look on how different is the microbiota of patients with metabolic syndrome if they have or they do not have type 2 diabetes. We have here just one plot to show something which I think is really important. This is a beta diversity of the microbiome. Basically, each square or circle represents a different patient, and we basically superimpose these types of microbiota, and we can see that they are actually quite similar. And this is really important because that means by giving this patient some probiotic or a certain diet, we can make this microbiota resemble the one of the healthy individual. And as I said before, this was done in London, but well, we wanted to do that here in Bucharest. So we had the infrastructure at our research institute and of University of Bucharest, but we did not have the reagents. So we had a small grant to actually buy these reagents and to do the experiments ourselves. And we did that last year. We have here the ion torrent machine. This is the process of sequencing, and this is us with our first microbiome that we sequenced. And of course, we're very happy. We can tell that from our eyes. We were wearing masks, of course. Of course, that's a big step for us that we can do that sequencing here at the University of Bucharest. And we actually will do in CVIS next year, a summer school regarding this microbiome sequencing and how we prepare the data and how we analyze them. So we are really, really happy that we are able to do this. And this is an example of how the data looks like. It's a little bit different. It's a different sequencing platform. We actually have these pie charts with the various abundance of various microorganisms. We have here the healthy individual, which has a lot of bacteroides. 37% of the microbiota is bacteroides, which has 6% Fecalibacterium, and we have some actinobacteria, around 3% of it, as part of the microbiota, and 12% Parasuterella. And if we look at patients with diabetic nephropathy, we have less bacteroides, but we have a lot of proteobacteria, both beta proteobacteria and Enterobacteriaceae. We have 7% of the microbiota represented by these Enterobacteriaceae, which are pathobionts. We have similar levels of Fecalibacterium, and we lose a lot of actinobacteria. So we do not have this bifidobacteria, which we see in the healthy individual. So why do we struggle so much? Okay, why do we need to know about these microorganisms? Well, we want to go from bench to bedside, and we want to improve this concept of personalized medicine, because at the moment each patient has the same diet and the same treatment and so on. And this is not the case, because each one of us is different and everybody has a different microbiota. So what we aim is to do the microbiome sequencing of an individual and to do also a metabolomic analysis. And we are also have underworked this type of experiments where we look at the metabolome of the host, and then we do a patient stratification, and then we do different intervention strategies, because we don't want to follow that concept of one size fits all, because that's not the case when we look at the microbiota. And then of course we want to do the patient follow-up and see how they respond, because in case of type 2 diabetes, our aim is not to cure diabetes, but to make these patients to have a better life and to have as less complications as possible. And again, here I listed some of our research, our reviews that we are related to this concept of from bench to bedside. And of course, because we have this ongoing pandemic, we are also looking at the SARS-CoV-2 infection and the impact of the microbiome in this infection. I have listed here a very useful review, which shows the fact that in the health condition, we have a lot of Eubacteria, Fecalibacterium prausnitiu, Roseburia, Lactospiraceae, which produce a lot of short-chain fatty acids. And in case of COVID-19, we lose some of these beneficial symbionts such as Eubacterium, Fecalibacterium, Roseburia, and we have an increase in pathobionts or opportunistic pathogens such as Clostridium or Actinomyces or Bacteroides nordi. And what we do now, and we are in the stage of collecting the samples from the individuals, is to look at the microbiome of patients who had COVID-19 and to compare the ones who had asymptomatic disease or mild disease or severe disease, and to see what changes are happening there in terms of microbiome signatures. And of course, with this, I want to thank all the people involved in these results presented here. There are a lot of teams and who have contributed one way or another to this data shown here. First of all, we have a huge support from University of Bucharest to do this experiment. Of course, we had the collaborators from London, from UCD, and we are still in contact with them, and all the hospitals who provide the samples. And of course, the funding agency who funded our research to study this on higher patient cohorts. And we started last year, and it's a little bit more difficult now to gather more patient samples because we have the COVID situation. And of course, it's more difficult than usual to get patient samples. And I want to thank you for your attention and to wish you to make microbiome friendly choices. So thank you.